Okay, so we come now to the next topic. It's all about the cell structure and function. As presented by the first discussant, so the prokaryotic cells differ in the number of major ways from eukaryotic cells. So lower eukaryotes, just like the protozoa, algae, and fungi, lack some of distinguishing features of metazoan cells. So nevertheless, all eukaryotic cells appear to be similar versions of the same overall plan. So in the past, some cytologists consider that bacterial cells were merely similar replicas of the cells of higher forms, and that the failure to distinguish comparable subcellular structures in bacteria could be attributed to the limitations of the microscopic and cytological techniques available. So this may be proved to be the case. So since many recent investigations with highly improved techniques and equipment have shown that there may be a closer similarity between eukaryotes and prokaryotes than previously thought. So it has also become apparent that the cells of Archaebacteria or Archaea display important structural differences from the cells of either Eubacteria or eukaryotes. So we come now to the eukaryotic nucleus. So eukaryotes or eukarya display a cytologically distinct nucleus, the organizational and regulatory center for virtually all of the biochemical and hereditary processes of the cell. With the aid of the electron microscope, it has been possible to demonstrate many structural details of the eukaryotic nucleus. So, in figure 7.1, it shows a mammalian cell with a well-defined nuclear membrane composed of at least two distinct layers. So, the outer surface contains pores with tubules that transcend both membrane layers. The amoebae, protozoa, algae, and fungi, the yeast and molds also contain discrete membrane-bound nucleus. So in 7.1, so this is the electron micrograph of portion of mammalian cell. So the region of this pancreatic exocrine cell between the nucleus, which is represented by a small letter N in the figure on the lower right, and the plasma lima on the lower left is occupied by numerous cisternae of the rough endoplasmic reticulum, or represented by rare in the figure, and a few mitochondria, which is small letter M. The prominent pores can be seen in the nuclear membrane. So the ribosomes appear as small black dots lining the rear. So the nucleolus is not visible in this photograph. So while in figure 7.2, this is the representation of fine structures of cells of lower eukaryotes. As you can see in the figure, the subscript letter A, that is the representation of the general view of a section of a cell of Saccharomyces cerevisiae. Prominent features include the cell wall, the nucleus with distinct pores in the nuclear membrane, mitochondria with cristae, and intermembranous structures. So, the subscript letter B, that is the fine structures of hyphal element of the fungus Mucorgenevensis. So, nuclei with pores, vacuoles, mitochondria, the endoplasmic reticulum, cell wall, and dark bodies are visible. Okay, next we have mitochondria. So mitochondria are subcellular organelles that carry out oxidative metabolism, or what we call respiration in eukaryotic cells. So the highly membranous interior of the mitochondrion contains the cytochrome system and oxidative phosphorylation processes that yield energy in the form of ATP. So lower eukaryotes, just like the yeasts, algae, filamentous, fungi, and protozoa have all been shown to contain mitochondria. So although their number and distribution vary widely from one cell to another, so even within a given species, the number of mitochondria per cell and their intracellular arrangement may vary with the cultural conditions and growth phase. So mitochondria possess two distinct membranes, the outer and inner membranes, which may be closely associated with each other at certain contact sites. So, the inner membrane is highly invaginated, forming plates referred to as cristae. So, yeasts produce mitochondria actively when grown in aerobic environment. So, under anaerobic conditions, mitochondria are not observed. Yet, upon shifting back to aerobic conditions, they quickly reappear. So, yeast cells grown under strict anaerobiosis contain mitochondria-like particles designated as promitochondria. So, these structures lack most of the characteristics components of respiratory chain but still contain ATPs, mitochondrial DNA or the mtDNA, and various structural proteins. So, anaerobic growth apparently induces a differentiation of yeast mitochondria. So, when yeast cells are grown in the presence of high concentrations of glucose, the respiratory and energy transduction system are repressed even under highly aerobic conditions. So the system responsible for mitochondrial gene replication and expression is completely blocked, and the cell satisfies all of its energy requirements by glycolysis. So we come now to the microbial cell surfaces. So in eukaryotic cell surfaces, this characteristics is not unique to microorganisms, nor it is strictly confined to either the plant or animal kingdom. So the cell walls of vascular plants are largely composed of cellulose, a B14-linked polymer of glucose, as shown in figure 7-9. So as you can see in the figure 7-9, this is the chemical structures of cell wall polymers. So cellulose, a B14-linked polymer of glucose, chitin, a linear polymer of N-acetyl-D-glucosamine, glucan, a branch polymer of glucose, and mannan, a branch of polymer of mannose. Okay, so the walls reveals the presence of the 29 B-glycans. Both 1,6-glycan and 1,3-glycan are found in 31 mannan and the 13% protein as shown in figure 7-9. So the mannan-rich outer layer of the wall contains a higher portion or proportion of 1,6-B-glycan. So yes, cell walls also contains small percentages of lipids and other materials. So chitin, the poly and acetylglucosamine is present in small amounts in vegetative cells of yeast, being confined almost entirely to the ring encircling the septum of budding yeast or the bud scar in the mother cells. However, the septum and the chitin ring encircling it shows a different chitin. So glycan ratio in the cell walls of all filamentous fungi so far examined. So chitin is the only common constituent found. Okay, so in the prokaryotic cell surfaces, in the chapter 1, it was stated that the high molecular weight sugar-containing a rigid structure called the peptidoglycan form the major backbone of the murine succulus of the cell wall of both gram-positive and gram-negative bacteria. Indeed, the production of peptidoglycan is considered to be the hallmark of the eubacteria. So the archaea produce a pseudomurine and associated with surface layer or the S layer composed of protein or the glycoprotein. So in many archaebacteria, The S layer may represent the only surface component outside the plasma membrane, as shown in figure 7.10.4. So, as you can see in figure 7.10, so this is the representation of the envelope profiles of conventionally fixed and embedded bacteria. So, bar equals a 50 nanometer, that is for number 1. Amorphous wall fabric of B. subtilis, having 168 arrow, lies directly above the plasma membrane. So, number 2 is Escherichia coli, possesses a thin peptidoglycan murin layer above the plasma membrane, over which lays a wavy outer membrane. So the region between the outer and plasma membranes is called the perisplasm or plasmatic space. So the waviness of the outer membrane is believed to be an artificial of the conventional fixation embedding technique. And much of the periplasm has been leached out. So number 3, in the figure, that is the Clostridium thermosaccharolyticum, is an example of a gram-variable bacterium that has a wall profile intermediate between the gram-positive and gram-negative formats in 1 and 2. So, the peptidoglycan layer is thinner than that of B. subtilis but thicker than that of E. coli. So, above this layer is proteinaceous layer of periodically arrayed subunits. So for the number 4 figure, as you can see in figure 7.10, that is the Metanococcus volti, is an archibacterium and possesses only a thin S layer above the plasma membrane and its sole wall layer. So we come now to the outer membranes of gram-negative bacteria. So the gram-negative bacteria display a prominent outer membrane that is peripheral to the perismatic region and peptidoglycan sacculus. So this outer membrane is covalently attached to the peptidoglycan layer through the lipoprotein and serves to reinforce the shape of the cell and to provide protective barrier against the external environment. So under the electron microscope, the outer and inner membranes present a similar appearance as a double track layers. So chemical analysis shows that there are similar in lipid content. Some components of the outer membrane are qualitatively similar to those of the cytoplasmic membrane. So however, the outer membrane appears as an asymmetric bilayer, with the external layer being composed of primarily of LPS, and the inner layer containing primarily phospholipids. So the outer membrane proteins of OMPS, called porins, form large water-filled pores with diameters of 1 to 2 mm that traverse the membrane and regulate the access of hydrophilic solutes to the cytoplasmic membrane. So as you can see in figure 7.17, so this is the representation of the diagram of gram-negative bacterial cell surface. So the ovals and rectangles, as you can see, that is the represent sugar residues. So circles depict the polar head groups of glycerophospholipids, and the MDO, membrane-derived oligosaccharides, and the core region of LPS is that the E. coli of K12, a strain that does not normally contain an O antigen, repeats unless transformed with the appropriate plasmid. So we come now to the cytoplasmic membrane. So the cytoplasmic membrane of bacterial cells is a bimolecular lipid leaflet of phospholipid molecules aligned at their hydrophobic ends. So the polar phospholipids are hydrophilic and face the external or outside of the membrane and the internal or cytoplasmic side of the membrane. So various proteins and other components of the cell may be partially or wholly embedded and the membrane layer, as you can see in figure 7 to 17, that the cell membrane serves as the permeability barrier, so preventing most solutes from gaining entrance to the cytoplasm except through specific transport proteins present in the membrane layer. So the phospholipids sa bacteria contain ester-linked phospholipids, whereas the archaea contain fatty alcohols linked to glycerol via either diether or tetraeder bands. We have also other membranous organelles. So under the electron microscope, certain organisms that have been shown to contain membranous structures other than the cytoplasmic membrane. So membranous structures that appear to be sac-like invagination of the cytoplasmic membrane of bacteria, termed as mesosomes, have been observed with the considerable regularity. So originally, a variety of physiological functions were attributed to mesosomes. So however, these structures have been shown to vary in size and appearance depending on the fixation procedures used in preparing cells for electron microscopy. So as a result, some consider these structures to be artifacts that can be manipulated by changes in fixatives or fixatives. So, and much of the earlier work on mesosomes had been discredited. So the Bertholles, these invaginated structures may represent functionally different membranous entities that will require further study before they are dismissed as artifacts of chemical fixation procedures. So, the marine bacterium, or what we call the Nitrocytis ocinus, possesses lamimar membranous organelles. These membranous organelles consist of approximately 20 vesicles to flatten the lumen in only 100 A thick. So the outer surfaces are in contact and form a triplet surface with an accentuated center line. So the lamellae almost traverse the cells, so displace the cytoplasm and the nuclear material and form the most prominent cytological feature of the cell. So we come out to the capsules. So many microorganisms produce an external layer of mucoid polysaccharide or polypeptide material that adheres to the cell with sufficient tenacity to be observed by simple techniques such as a negative staining within the ink in figure 725. With this outer layer of materials which is sufficient size to be readily visible, it is referred to as capsule. So several examples of well-defined capsules are shown in figure 725. So many microorganisms produce a variety of extracellular materials that may be too sparse or too soluble in the medium to be observed by microscopic techniques. So these materials are often referred to as slime layer. So the production of capsules or slime layers may impart the ability to form biofilms that enhance their ability to attach to surfaces. Okay, so the last topic to be discussed under the structures, cell structures and function is the pili or Fimbriae. So the bacteria from many different genera produce a multiplicity of non-flagellar filamentous proteinaceous appendages called pili. So the Latin pilus meaning hair or Fimbriae, Latin fimbra meaning French, conjugative for F. pili, are involved in the mating process described in chapter 3. So a number of other types of pili, sometimes referred to as common or generalized pili, so are also produced by numerous bacterial species. So these pili play a role in adherence of microorganisms to surfaces or to specific receptors in eukaryotic cells, aiding the colonization of these ecological niches through the formation of biofilms. So the type of 4 pili are involved in the form of motility known as switching. So members of Enterobacteriaceae produce a wide assortment of pili or membranae. Prevalent classes of Fimbriae are of type 1. So these pili are composed of short-tipped fibrillar structure containing film G and the film H adhescent attached to a rod composed of film E subunits. So the film H adhescent mediates binding to mannose oligosaccharides. So the mannose sensitivity. So chaperone usher pathway, a donor stand complementation mechanisms in which the chaperone donates strand or complete pilin formation mediates phylosynthesis. So expresses of the type 1 fimbriae in E. coli exhibits recombinations mediated on and off phase variation. So the phase variation correlates with the orientation of a short invertible 314 bpDNA element or switch located immediately upstream of FIM-A or fimbriae. So the promoter for FIM-A is thought to reside within the invertible element and to direct the transcription of FIM-A when the element is one orientation but not with the element in the alternate orientation. That's the end of my presentation. Here are my references. Thank you everyone for listening. Have a good day.